This mini lecture covers the organelles that aren't directly involved in making proteins. Cell membrane is sometimes included as a protein-making organelle. There's an entire other mini lecture on cell membranes. So just a little review. Remember, it separates the internal from the external, right? The skin is to you as a cell membrane is to a cell. What you see here, I don't know if you know what you see here. Some of you might be like, ooh, I know, is adipose tissue. And we use this pinkish purple stain, and the lipid-based cell membrane takes up this stain. So this is covered in another mini lecture in great detail. What is inside a cell? Well, if you were to peel open the cell membrane, you would see a lot of gooey, watery substance, and then you would see little organelles and other things inside the cell. The gooey substance is called the cytosol. That is the word intracellular fluids. So ICF refers to the cytosol of all of the cells in your body. When you're talking about one cell, you can say that the internal fluids are the cytosol. So what's in the cytosol in general? What's the most common things? Lots of proteins, tons of proteins negatively charged, lots of amino acids, because your cells really are protein factories. So there's tons of amino acids. And interestingly enough, I just want you to remember there's lots of potassium. I don't know, connect it with a banana. The cytoplasm is a word that is more often used when describing the contents of a cell. In fact, I don't think that your book uses the term cytosol ever again. I don't. I will say cytoplasm. The cytoplasm is all of the fluid, so it is the cytosol, but it's all of the organelles in there too. The nucleus, the endoplasmic reticulum, all of the organelles. All right, because this lecture is not protein making organelles, this is like a random collection of other organelles that just exist. So I try to start from like the outside in. The first couple slides describe structures that are on the outside of a cell, superficial surface. This cell up here, right in the top middle, is a black and white picture of a cell. And let's say that this cell is from the small intestine. There are all of these little projections on the cell, which are called microvilli. These are not hairs, right? Think about hairs are like attached to your body, but your fingers are part of your body. Does that make sense? That's what a microvilli or a villus is. What are these for? They're for increasing surface area. This cell wants to suck up as many digestive nutrients from your small intestine as it can. And these little projections or fingers allow them to have more surface area to move things across. So these are common in the small intestine, and just remember, not only in the small intestine, but they're very common in those cells. Microvilli get confused with cilia. So cilia are very, very thin extensions of the membrane. So they might not be like fingers, but thin fingers. I don't know. So they are extensions of the membrane, but they're very, very fine and very, very thin. And what they do is they tend to all beat together, kind of like people at a stadium doing the wave. They all beat together, and you can see that down on the bottom left. We have some cell surfaces with cilia, and they all are kind of rhythmically beating in a pattern. And what happens in your respiratory system is that these beat, moving mucus superiorly into your throat. And then you cough, swallowing it down into your esophagus. And this is what's called the mucociliatory escalator. You also have cilia in reproductive tracts. The uterine tubes have cilia that move ovulated ovum to the uterus, And the vas deferens, now called the ductus deferens, also has cilia in it to move sperm along. I don't have much to say about the cytoskeleton, and that's unfortunate because it is we're discovering very fascinating things about it. But I don't have much to say because you can guess from the name of it what it does. It is an internal structure, an internal network. What is it made of? It's made of proteins. And the three different types of proteins, which I don't require you to know, they're listed down here: actin, microtubules, and intermediate filaments. And they support cells in different ways, right? So more actin filaments can make a cell more flexible, more microtubules, you're more rigid. So different proteins, different purpose. But what we're finding is that the cytoskeleton is not just bones used for support. It's actually a transport highway. It's like a network where the cell can move proteins from one part of the cell to another. And we're starting to understand that that's what the cytoskeleton does. These two items will become more important to us when we look at mitosis, which is the division of a cell. So there are these things called centrosomes, and a centrosome is a pair of centrioles. I'm sorry. When a cell isn't dividing, the centriole, they hang out as a centriole. It's like a pair of these. And it's needed for reproduction, but it hangs out as the centriole when it's not being used for reproduction. When reproduction starts to happen, then the centrioles will split up into their individual centrosomes, and we do mitosis. So this is needed for reproduction. Interestingly enough, many of us know that neural damage doesn't heal very well, especially traumatic brain injuries. One of the consequences of that is that neurons don't have these structures, or they don't use them effectively, but these are cells that don't divide. They don't go through mitosis. It is the original cell in your body from the time you were born, and they don't have the machinery to divide. There is only one type of cell from humans that have a flagella. Flagella is plural, flagellum is singular. Sperm are the only human cells that have this. And this is a tail, so it's not projections like microvilli, it's not hairs like cilia. This is a tail. And you might think that it waves back and forth. It does not. It rotates. So it took us a little bit to figure out that this has rotary motion. And you can see the mechanism here that allows the tail up at the top to spin around in a circle. The tail can be damaged by heat. So holding a laptop over your lap, if you're male with active testes, wearing underwear that is too tight, bringing the testes closer to the body's heat, can damage the development of the flagella, causing a decrease in reproductive viability. I hate it when students and books say that mitochondria are the powerhouse of the cell. I don't know what that means. To me, mitochondria make ATP. Boom, that's what they do. They take oxygen and glucose in your blood, and they they make ATP from oxygen and glucose. And the waste products are water and carbon dioxide. ATP have an internal membrane. It's like a little bean, and has an internal membrane, and the internal membrane has tons of folds in it to increase the surface area. And in these folds are millions of enzymes. And these enzymes take part in making ATP. Cells that need more energy, like muscle cells, they have more mitochondria. Your neurons have tons of mitochondria. I mean, they use glucose all the time as you're studying. So when cells are more active in what they do, like muscle cells, they will have more mitochondria. Lysosomes are pretty, I find them kind of fascinating. I don't know why. They're very simple. It's a vesicle. A vesicle is like a bubble within a cell. I described them in the cell anatomy lecture. So a lysosome is a bubble, but in it specifically are digestive enzymes. So this will float, it's like a little mobile stomach. This will float throughout the cell, and if there's damaged organelles or if there's something that needs to be removed, the lysosome will take it in, break it down, and then release its products to be used again. There is a very distressing disease called Tay-Sachs, which is genetic. We do test for it at birth. You can before if there are indicators in your family. But Tay-Sachs has to do with the inability to make one enzyme in lysosomes of cells. And because of this, there's a specific lipid that's incapable of being broken down. So over time, the lysosomes accumulate more and more and more of that lipid. And then they get engorged. They grow, pushing out other organelles in the cells. And they do this to every cell in the body. Students often confuse lysosome with peroxisomes. So lysosomes are in many different cells, and they kind of digest and break down, normal substances that have run through their life, like machinery that you throw away. Peroxisomes, on the other hand, they are vesicles, but they have a specific purpose, or more specific, and they're full of hydrogen peroxide. Look at this picture up here on the right. Like, this is kind of fascinating. You can see the folds of the rough ER. You can see a mitochondria, and you can see the folds in it as well. And then, of course, you can see all of these peroxisomes that are darker in color. Down here on the bottom right, the peroxisomes are releasing their hydrogen peroxide, and you can see that dark color leaching out from the cells. So this is a good word, a phagocyte. Phago means to eat. This is a cell that eats other cells, basically types of immune cells. So phagocytes will engulf a pathogen, a virus, a bacteria, a fungal pathogen, any kind of infection, and it will use its peroxisomes to kill that invader. And then it'll usually bring bits of that invader to somebody else in your immune system and say, I found this. Should we launch an attack? I've included this picture here in this PowerPoint as well as the other one. I like this picture. It's from another book. It's not as complicated as the one that's in your book. It does have all of the important stuff in it, and it's very well illustrated too. So I just wanted to show you this. I think that this is the picture that appears on many things like quizzes or Q&A assignments that we might have in the class.